There's the equation for cellular respiration again. By the end of glycolysis, we've made 2 pyruvate ions, and so we don't have to keep track of positive charges and negative charges, we can think of this as 2 molecules of pyruvic acid. But we haven't used any oxygen yet. We haven't made any carbon dioxide yet. We haven't made any water molecules yet. Apparently, we're not done yet. Well, if you look at pyruvic acid, it still has some carbon-hydrogen bonds, which means it'll burn, which means it still contains some usable potential energy, so no, we're not done yet. Well, this part looks like it's ready to become carbon dioxide, so maybe that's what happens next. If we take away carbon dioxide, we're left with 2 carbon atoms, 4 hydrogen atoms, and 1 oxygen atom. To turn these last 2 carbon atoms into carbon dioxide, we'll need 4 oxygen atoms, and right now we only have 1 oxygen atom. Aha, you say! Here's where we start using those oxygen molecules from the balanced chemical equation we looked at earlier. Nope. We're going to get the 3 oxygen atoms we need from 3 molecules of water. Wait, what? I thought we were going to make water molecules, not use water molecules. We're finally going to use the oxygen molecules and make the water molecules at the end of the electron transport chain. Okay, fine, we added 3 water molecules. Now we have 2 carbon atoms, 10 hydrogen atoms, and 4 oxygen atoms. If we turn the 2 carbons and 4 oxygens into 2 molecules of carbon dioxide, we're going to have 10 hydrogen atoms left over. Here's an idea. Let's squeeze the remaining usable potential energy into high-energy electrons, and then let's use these 10 hydrogen atoms to carry away 10 high-energy electrons. Well then, that sounds like a plan. We're going to run pyruvic acid through the Pyruvate Dehydrogenase Complex. That'll pull off a carbon dioxide molecule and carry away 2 high-energy electrons using our old friend NADH plus H+. Pyruvate dehydrogenase then takes what's left, the 2-carbon fragment, and sticks it onto coenzyme A, making acetyl coenzyme A. So what the heck is coenzyme A? For now, we can just think of coenzyme A as a tool for conveniently handling a tiny 2-carbon fragment. Here's a potentially weird analogy. If you're polishing a diamond on a big, spinning, abrasive wheel, you do not want to hold the diamond in your fingertips because you don't want to have your fingertips torn off. So you attach the diamond onto the end of a tool of some kind. Coenzyme A is the tool that gives the citric acid cycle something to hold on to so we don't have to worry about these relatively small intermediates floating away. Here's the citric acid cycle, or as I sometimes call it, the giant wheel of chemical pain and suffering, with all of the chemical structures shown so small that you can't really see anything. That's fine, it'll all be fine. The citric acid cycle is going to add the 3 water molecules we just talked about in steps 1, 6, and 8, though the water molecule in step 6 is hidden as part of the phosphoric acid. We'll talk more about that in a minute. We remove the 3 carbon atoms that came into the party in the pyruvate ion, making 1 molecule of carbon dioxide in the pyruvate dehydrogenase complex, and the other 2 molecules of carbon dioxide in steps 4 and 5 of the citric acid cycle. We send away a total of 10 hydrogen atoms, carrying 10 high-energy electrons to the electron transport chain, 2 from pyruvate dehydrogenase, and 2 each in steps 4, 5, 7, and 9 of the citric acid cycle. Every step of the citric acid cycle does something interesting, or at least potentially interesting. Step 1 sticks the acetyl group of acetyl-CoA onto oxaloacetic acid, turning one thing with 2 carbon atoms and one thing with 4 carbon atoms into one thing with 6 carbon atoms. Steps 2 and 3 rearrange the citric acid to make it isocitric acid instead, because that makes it easier to pull off carbon dioxide molecules in steps 4 and 5. Step 6 does substrate-level phosphorylation, just like we talked about in glycolysis, except for some reason this time we're making GTP instead of ATP. Step 7 is interesting for several reasons. First, step 7 puts its high-energy electrons onto FAD instead of NAD+. Secondly, The enzyme complex that catalyzes step 7 of the citric acid cycle is also the second respiratory complex in the electron transport chain. Third, the high-energy electrons on FADH2 are transferred almost immediately onto coenzyme Q10, which then sends them on to the third respiratory complex. The high-energy electrons on FADH2 therefore skip the first respiratory complex, which is why the high-energy electrons on FADH2 cannot make as much ATP as the high-energy electrons on NADH plus H+. Surely that's enough for now, right? Well, I also made slides for each of the individual steps, so here they are. Pyruvate dehydrogenase, step 1 of the citric acid cycle, steps 2 and 3. Note that the enzyme aconitase catalyzes both of these steps. Step 4, and then rotating around a bond in alpha-ketogluteric acid so it looks the way we're used to seeing it. Step 5. Step 6. Here I'd like you to count the atoms in GDP and GTP to convince yourself there's a water molecule hiding in phosphoric acid. Step 7. Step 8. And at long last, step 9. With one more bond rotation to make oxaloacetic acid look right. Okay, now we're done here. Next time we'll discuss the electron transport chain and oxidative phosphorylation. In the meantime, thanks for listening. I'll talk to you later.